Yeah, so I got my PhD in health, sport, and exercise sciences from the University of Kansas with a focus on exercise physiology. My dissertation here was largely focused on neuromuscular adaptations to resistance training. And while it's not a complete direct carryover to some of the bone research we're doing now, it carries over to some of the other research I'm doing. So currently working on a proposal to analyze the effects of different rehabilitation strategies to improve neuromuscular function in veterans following stroke. So there's a direct carryover there in terms of the techniques and methodologies that I learned here under Dr. Trent Herta. On a more general perspective, a large part of the PhD is just learning how to conduct research properly, and I think all aspects of that directly carry over to everything I do on a daily basis. So right now at the University of Pittsburgh, we have a big project that is interested in better understanding all of the beneficial adaptations that resistance training and exercise can have on bone formation. One of the biggest issues in the military right now is the high prevalence of stress fractures. It's one of the biggest causes of downtime in the military. I believe it's the second most prevalent musculoskeletal injury in females and the fifth most prevalent musculoskeletal injury in males. So they have a big interest in doing everything they can to reduce the number of stress fractures that people experience. As a result, we are examining the effects of exercise and loading on bone a number of different ways. So this is a big DoD study that has two big arms. One of the arms is investigating elite athletes 6 months apart, including their competitive season, and we're looking at the bone adaptation that occurs over the competitive season in athletes. The other arm has a resistance training group, a control group, and then an ROTC group. In the resistance training group, we went back to the literature and analyzed all of the things that seem to maximize the osteogenic or bone benefit of resistance training. There are well-established training programs for muscle hypertrophy, for strength, for power, for aerobic fitness, but the ideal optimal way to train bone is not really determined yet. So we went back to the literature, looked at animal research, human research, and came up with a training program that we think is going to maximize the osteogenic benefit. So we're doing that training at Pitt. We also have an active control group so that we can measure our training versus a control group. And then because it's a DoD study, we want a military standard of care. And that's where the ROTC comes into place. We are testing the University of Kansas ROTC cadets. We have 2 visits. Initially they come in, we familiarize them with all the different testing procedures. At the second visit, they come in and they complete 2 different types of bone imaging. We use a DEXA, which for a while has been the gold standard of bone mineral density assessment. So they have a DEXA scan of their total body, hip, and spine. From the DEXA, we can also determine their body composition, the relative amounts of lean body and fat mass that they have. And we're also using a state-of-the-art high-resolution peripheral quantitative computed tomography, the HRPQCT, which is a newer version of bone imaging. It's higher resolution. It allows us to look at the different types of bone, namely cortical versus trabecular bone, at both bone mineral density as well as bone microarchitecture and geometry. This allows for a much better prediction of the failure, load, and stiffness of the bone and allows us to better predict fracture risk. Aside from the bone imaging, we will also be taking blood samples at rest and following an exercise bout. The reason that we're taking the bloods is that we not only want to understand how the ROTC training, how our laboratory-based training affect the bone in terms of the imaging outcomes, but we also want to get a better understanding of the different physiological mechanisms underpinning those improvements in bone. So we know that there are a number of different hormones and signaling factors released from a variety of tissues in response to exercise, which have downstream effects on the bone. And so with the blood samples, we're able to look at myokines, adipokines, osteokines, as well as an interest of our lab is extracellular vesicles, which can carry cargo from these different tissues to distant targets and elicit adaptation. I think this is a first step in trying to combat the stress fracture problem. We know that in the military, the issues that contribute to the increased stress fracture risk is multimodal, multifaceted. It's things like the high volume, which in some people is very different from what they had done prior to basic training. It's the insufficient sleep, caloric intake, as well as NSAID use that people use to combat the soreness associated with training. With this, if we could determine a very osteogenic or training program that's beneficial to bone, the next steps would be to investigate the role of these different factors, sleep, nutrition, NSAID use, on the adaptations we see to this training program. I think it's a great study. It's a great opportunity. From what I've seen, it's one of the most in-depth and comprehensive analyses of osteogenic adaptations to training. So we're employing a lot of state-of-the-art techniques, and some of the early results are already really promising. I can't take all the credit, but I did have a big hand in writing this proposal along with the director of the Neuromuscular Research Lab, Dr. Brad Nindle, and my colleague, Kristen Colton.